Welcome to Study Loop.
Hello students, the given question has asked in different parts. In the first part of the question, the non-template DNA sequence is given and we have to write complementary DNA sequence for it. So this is non-template DNA. The key point is that while writing the complementary DNA, you should know that this nucleotide base pair, they will pair with their complementary nucleotide base. That is, A will base pair with T and C will base pair with G and vice versa. That is, G will pair with C and T will pair with A. So the complementary strand will be, so here you can see there is G in non-template DNA. So G will pair with C, T will pair with A will pair with T will pair with A and goes on. So here you will find every base pair has its complementary base pair. This is how complementary DNA sequence is. In the next part, here again a template sequence of DNA is given and we have to write the mRNA transcript for it. So this is template DNA sequence which is given. In mRNA, the only thing is that this T will get replaced with U because in mRNA, whether it is mRNA, tRNA or rRNA, you will find U instead of T. So U will pair with A and not T. So let's write mRNA transcript. So here this is mRNA and template DNA sequence. Here you can see that T base pair with G, T will base pair with A will base pair with U and not T. That is the difference. So this is how the mRNA sequence will be. Next part of the question. In this next part of question, we have to write down the amino acid which will be coded by this codon. So the here first codon is the AUG, that is start codon, which will code for methionine. Next amino acid is glutamine. Then you will find leucine, arginine, serine, arginine, tyrosine, aspartic acid, serine, arginine, leucine, alanine, and lastly lysine. So this is the polypeptide chain and this is the each amino acid which will build up, and this is the peptide bond and this will result in the formation of protein. I will write amino acid, yep, polypeptide chain. Next question. In this question, we have asked to write the sequence of a mutated template strand that has a single base mutation, that is point mutation, that would result in nonsense mutation. So let's change the base pair. So let's say here this UCG which codes for serine, here this C will get mutated and here another base pair will add, that is A. So it will be AUG, that is nothing but stop codon. So mutated template strand would be, so here you can see UAG is there over here and this C has been mutated, that is point mutation. Now this will do not code for any amino acid, but it will stop the process of translation. So the result transcript will be, or you can say the amino acids will be methionine, LAU, arginine. and it will be stopped. Further process will stop. So end. This is how mutation affect and it entirely change the nature of protein as one base pair will get mutated. This will result in wrong amino acid insertion. So here it will lead to the inhibition of process of translation and thus it will affect the nature of protein. I hope you found this video helpful. Thank you.